Okay. I think I'm okay. Okay. Okay, we need to finish chapter 6 real quick. And then we're going to get into chapter 7, which is the structure of the cell membrane. Sorry, I'm making sure the volume is on. Nope, I don't need that. Am I muted? Am I not muted? No, I'm not muted. Okay. Okay, so we left off on Friday, and I just want to rehash where we are. We were talking about this last part of the chapter talks about extracellular structures and how cells are connected to each other. So we talked some about the plant structures that are here that we hadn't talked about necessarily in detail, like this big blob in the middle is the what? Central vacuole, which helps to maintain cell shape by keeping the water balance okay. This green thing is a chloroplast, which converts light energy into chemical energy. And this is labeling the cell wall of two different cells. So showing you how the cells fit together. Here's the cell wall of one cell, the cell wall of the other. In between that cell wall, we did not mention this. That's an M. Is a layer called the middle lamella. This is made up of sticky polysaccharides that help hold the cells together, known as pectins. Has anyone here ever made jelly? You use pectins to thicken jelly. It makes jelly thicken. That's where it comes from. It comes from plants, from the layer between the cell walls. I thought that was kind of cool. And we talked about this briefly. These channels, these openings that are between plant cells, remembering that the membrane is continuous so that cells can share stuff in between each other. They can share nutrients, they can share ions, all that stuff. The environment between multiple plant cells can be the same because of those plasmodesmata. And you looked at this slide, this picture in lab, and you can see all these little channels that are connecting. This is a persimmon. Those are plasmodesmata, which are holding cells together. Why do we need to hold cells together and let cells communicate with each other? In a multicellular organism, are cells functioning independently? No. So they need a way to be held together. So we need to have these cell junctions, and the cells need to be able to communicate with each other to make a multicellular organism function. When we take a group of cells and put them together, what are they called? When they work together, we talked about this way back in chapter 1. A tissue, and then we hold tissue together to make organs. Okay, so these cell junctions that we're talking about, plasmodesmata, and what we're going to talk about with animal cells, are all ways to hold cells together to make tissues, and tissues together to make organs, to make a functional multicellular organism. So again, that plasmodesmata are plant cell. Those are the only plant cell junctions we're talking about. We're going to talk about 3 different animal cell junctions. So 3 different ways that animal cells can be held together. And the first one is what is called a tight junction. Guess what this means? The cells are really tight together. Okay, not a complicated name for this one. This is just when the plasma membranes are held together so tightly with proteins that there's a continuous seal. So it produces a continuous seal between the cells. So there's not really any space between them. It's tight. So the plasma membranes are held together with proteins producing this continuous seal. That does not look like the word seal. An example of this is just your skin. Your skin, the epithelial cells in your skin are held together with tight junction that makes your skin waterproof. And you'll see that's like a more, that's a little bit higher up, maybe, I guess I would say. It's a little higher in the layers of the skin. or whatever tissue we're talking about. A second kind of animal cell junction is a desmosome. And desmosomes are held together by one of the three kinds of cytoskeleton fibers that we talked about. So these are held together by intermediate fibers, so parts of the cytoskeleton. And this is what we call an anchoring junction. So I think your book uses the chart, it's like rivets, like a bunch of screws going through, holding it together. So it's not as tight, but it's anchoring, holding the cells together. And this kind of junction is the junction that you might find between muscle cells, holding your muscle cells together into muscle fibers and into larger muscle groups. And then the third type of animal cell junction are called gap junctions. This is what we call a communicating junction. And these occur where there are proteins. Protein starts with the P. Yep, I can spell today. Proteins are making channels. What does that sound like? What kind of other kind of junction does it sound like? That we just talked about on the last slide. Previous slide. The what? I'm sorry, I'm clicking the wrong one. Yeah, they're like plasmodesmata. So they're channels between cells. Again, this is allowing cells to pass materials between them so they can share materials. So it could be things like ions that are necessary, sugars. But the cytoplasm of the two cells is connected to share those materials through these protein pores. And these kind of junctions we would find in heart muscle specifically. The other one is more like this, when we say muscle cells here, we mean more like skeletal muscle. And a lot in embryonic tissues as they are developing. There's a lot of communication between cells. Yes, these are all animal cell junctions. So all different ways that cells can be connected to each other. And I meant, you hopefully saw some of these on the cell models in lab when you looked at the cell models. I meant to bring the animal cells, those show them as well. And I forgot to bring it. Right. So that's talking about cells being connected to each other. Cells, animal cells are also connected to extracellular structure. So we said the cell wall is like an extracellular structure for a plant cell, but animal cells don't have that to provide that support and structure. Animal cells are instead connected, a lot of them are connected to this extracellular matrix. Which we abbreviate as ECM because it's a lot shorter. So the extracellular matrix is for animal cells kind of like what the plant cell wall is for plants. This provides structure and support for cells that don't have a cell wall. So it's made up of lots of, I'm sorry, I'm writing this the wrong way. Glycoproteins What does glyco make you think of? Carbohydrates attached to proteins, glycoproteins are just carbohydrates covalently bonded to proteins. So these are proteins that are secreted by the cells that have carbohydrates covalently bonded to them that provide some structure and support, some places for cells to anchor themselves. A good example of this is collagen. Collagen is a connective tissue protein. That can make up to 40% of the protein in your body can be collagen. So it's a significant amount of protein in your body that is collagen. Again, it's not in the cell, it's outside the cell, it's extracellular, but it provides this structure. And these collagen fibers are embedded in this proteoglycan complex. And a proteoglycan is just a protein with all these carbohydrate chains coming off it. So it's got a core protein. And then all these are carbohydrate chains. And it can have so many carbohydrates that it's up to 95% carbohydrate chains. So all this is outside the cell. Then the cell manufactures these proteins like fibronectin. Fibronectin is the protein that's actually connecting the cell to the extracellular matrix. And again, it's a glycoprotein. And it attaches to this protein, which you'll see is embedded in the membrane, which we'll talk about more in the next chapter. And this is the attachment point. So the cell itself has integrin in its membrane. The fibronectin is connected to that, which is then connected to the collagen in the ECM. Again, providing that stability and support, that anchoring for the cells. And also allowing communication between the cells and the extracellular matrix. There's some stuff in the extracellular matrix that the cell might want. And so being able to have communication between the extracellular matrix and the cell cytoplasm is important. Pretty exciting. You guys seem overwhelmed with enthusiasm about the extracellular matrix. Me too. Okay, that is the end of chapter 6. Now let's see if I effectively shared both screens. I did. So we're kind of getting more and more detailed oriented about the cell. Okay, we spent chapter 6 talking more broadly about cell structure. Now we're zooming in just specifically on the cell membrane itself and looking at the structure of the cell membrane and how the cell membrane functions and why do we care about the cell membrane function. I assure you, you want to care about the cell membrane function. Even if you don't want to, you want to. And again, remembering whenever we talk about something in biology, structure determines function. So the structure of the cell membrane is going to determine how it functions. We've kind of been alluding to and talking about some things with the cell membrane. This is where we're going to get really detailed about it. This week in lab, you have a lab exam, right? Yeah. Next week in lab, you're going to be doing a lab with cell structure and function, looking at the membranes structure and function. So it's a really cool lab with beads and looking at what happens when you do different things to the beads, what happens to the membrane. It's one of my favorite labs. Okay, what kinds of macromolecules do we already know are part of the cell membrane because we've already talked about it before? What kind of macromolecules did we talk about in chapter 5? Yeah, it was 5. So one of the kinds of membranes that are found in one of the kinds of molecules that are found in membranes are lipids. And what specific kind of lipids did we talk about are really important? There's actually 2, but we talked about one more than the other. They do have hydrophobic characteristics. Yeah, so phospholipids are, oh, hello, don't do that. Phospholipids are one kind of lipid. Anyone remember the other one? We talked about it briefly, but not a lot. Cholesterol is another kind of lipid that's found in the cell membrane. So that's one category. What's the other category that's important as part of the cell membrane? Not the proteins. And so we'll be talking about a little more about phospholipids and their structures and how they affect the structure of the and function of the membrane, and some more about proteins. This is not to say carbohydrates aren't involved at all, but they're not as structurally important. They have important roles in the cell membrane, but not as structurally important. So when we talk about the cell membrane, we use the fluid mosaic model. Okay, when we talk about a model, means this is the best way we know how to describe it. Do we know that it's perfect? No. Do we change our minds if we learn more about the cell membrane? Yes, but this is the current working model of how we understand the way a cell membrane works. And you should recognize some of this because we just talked about it. And we've talked about it before. So these guys with the head and 2 tails are the phospholipids. So we're calling it a mosaic. If you've seen a picture of a mosaic, there's a picture in your book of a mosaic of a bird or something. You know, it's just a lot of different pieces put together. Okay, so one of the pieces of our mosaic are the phospholipids. Then we have these things that are embedded in it. These are our proteins. And then you'll see these, anytime you see a ring, chainy ring structure, what does that remind you of? Somebody said something, but quietly enough, I didn't hear it. A bunch of rings. Carbohydrates. So this is showing you here we've got a lipid with some carbohydrates attached to it. That's, my blue isn't showing up. A glycolipid. Okay, again, glyco should make you think of carbohydrate, and it's attached to a lipid, so it's a glycolipid. And then over here, this is showing a protein that's got some attached carbohydrates. That would be a protein with attached carbohydrates. A glycoprotein, yeah. Not a trick question. So again, depending on the function, the kind of cell that it is, there might be different amounts of proteins. Some cells might have more proteins than others. Some cells might have more glyco tags on them. But we've got this structure that should look familiar at least somewhat by now. Brandon, you mentioned that part of those molecules were hydrophobic. Which part of the phospholipids are hydrophobic? The head or the tails? The tails, so this part is hydrophobic. And then the heads themselves, the phosphoheads, are hydrophilic. So this membrane will self-assemble with the hydrophobic tails on the inside and the hydrophilic heads on the extracellular side and on the cytoplasmic side, because that's where the water is. So in here, these tails are held together by a weak interaction. That's just a hydrophobic interaction, which basically means they are trying to stay away from the water, so they stick together. It's a weak interaction, but it's still an important interaction. It's one of the side chain interactions that can happen in tertiary structure of proteins as well. So this is our working model. It's fluid and it's mosaic, and we'll talk about both of those components, but we're going to zero in again a little bit more on these structures. Again, we've already talked about this. These are hydrophilic. These are hydrophobic. What do we call it when a molecule is both hydrophilic and hydrophobic in the same molecule? Somebody said it. Yeah, so these phospholipids are amphipathic, which again, amphipathic just means it's got a hydrophobic part and a hydrophilic part together. The fluidity of the membrane has a lot to do with the phospholipids. What do we mean when we say that it's fluid? Yes, it can move, stuff is moving around in it. So we've got these phospholipids and these proteins, but they're not static. It's not just like we line it all up and it stays that way. So there's movement of the different molecules within the membrane structure itself. So it's not static. Proteins can move from one part to another. Lipids can move from one part to another. Generally, if a phospholipid, wow, that went a lot at one time. If a phospholipid is in this layer, it's not generally going to move to that layer. It doesn't switch, but it can happen. It's just more rare. What kinds of parts of the cell might help to move things around in the membrane? That's not a very well-worded question. It's not necessarily an organelle. When we talked about at the end of the structure of the cell, we said that there were things that could help move organelles and things around. Which parts of the cell did we talk about that help to move organelles? Still not coming up with good wording. Flagellum will move the whole cell, yes. But within the cell, we said we can move vesicles, yes. So we can use vesicles to move materials around. I'm not asking it particularly well, Trevor. Yeah, so the cytoskeleton, those parts of the membrane can be moved by the cytoskeleton. As the membrane is synthesized in the ER, it is moved out to the moved out to the membrane by vesicles. Did anybody watch the 21-second video of the motor protein moving the vesicles around? Wasn't it cute? Okay. 21 seconds of your life you can find between now and Wednesday. Watch the motor protein moving stuff around on the cytoskeleton. So the cytoskeleton can attach to different parts of the membrane and move those parts around as needed. So it can help hold them in place, and it can also help move them around. So what can affect the fluidity of the membrane? I feel like I need another slide. Did I make myself? No, I didn't. Okay. That says fluidity. So the membrane is sometimes more fluid and sometimes less fluid because of a variety of factors, some of which you'll investigate in lab next week with leads. Yes. How well the lipids are compacted, which is affected by their structure too. Okay, so what do you notice about this membrane versus this membrane? These tails versus these tails. Yeah, so these tails have that kink in it. Remember, that happens because of what? This kink in the tail happens because right in here, we've got that C double bond C. Because it's not saturated. Remember, saturated fatty acids have only single bonds because they're only bonded to hydrogen, but this is unsaturated. It's got that C double bond into a C, which puts a little kink in the tail. Because there's a little kink in the tail, that makes a little bit of membrane space, a little space between the phospholipids, and that makes the membrane more fluid. It's able to move around more because it's got a little bit more space because of these unsaturated fats. Over here, these are all saturated, which means we have only C double bond C in these long hydrocarbons. So these are just C's bonded to C's with H's sticking out the sides. There's not as much space, so it's more viscous, we say. It can't move around as much because it's more tightly packed. So the saturation of the fatty acid tails affects how fluid the membrane is. Okay, what else might affect? Temperature, definitely. The temperature can affect the fluidity of the membrane. As things get warmer, what happens to the atoms and molecules in them? They move faster. As they get colder, they move more slowly. And so they're not going to be as fluid when they're colder. They're going to be more fluid when it's faster and they're moving around a lot more. So a higher temperature means higher fluidity. To a degree, but we have an exception, kind of, sort of, not exception, a buffer against this. And that's these molecules. What are these molecules embedded in this membrane? Those are the cholesterols. Remember the 4 fused rings? So the amount of cholesterol can affect how fluid the membrane is. More cholesterol means less fluid generally. But cholesterol is also affected by temperature. So temperature has an effect on, not on the amount of cholesterol, but on how the cholesterol works. So cholesterol provides stability at higher temperatures. So if you have a membrane that's exposed to a higher temperature, that cholesterol is actually helping to prevent it from being more fluid. It provides some stability to prevent fluidity. So at higher temperatures, cholesterol makes it more stable. But at lower temperatures, it actually prevents these tails from getting super packed. So it actually helps it stay a little more fluid. So it's, some people call it a buffer. Remember how a buffer resists changes in pH? It's like a fluidity buffer. At high temperatures, it prevents it from being too fluid, and at low temperatures, it prevents it from being too viscous, too rigid.
What did you raise under?
Oh yeah, sorry, that's prevents packing of the tails. It prevents the tails from being all smushed together. So at higher temperatures, it makes it more stable, so they're not so fluid, too fluid. And at lower temperatures, it prevents them from packing too tightly, so it keeps it a little more fluid. So what do we get from this? Cholesterol is a good thing. Go eat some red meat and fried foods, and it'll help keep your cell membranes in better shape. No, I'm sorry. Your body makes enough cholesterol for you to take care of your cell membrane needs. You don't have to eat it. But it does have a good function. So fluidity. What do we care about fluidity? The fluidity of the membrane overall will determine permeability So if a membrane becomes more fluid, what do you think that happens? What do you think happens to the permeability? It increases. That can get to the point where it's problematic and it can make a cell membrane leaky. So too much fluidity is a bad thing. If it becomes too not fluid, if it becomes too rigid, what's that going to do to permeability? It's going to decrease it. Why is that a problem? Then stuff can't get in and out. Okay, we need the membrane to be permeable, but we need it at the right level of permeability. So membrane fluidity is really important because it helps the cell be able to control what's going in and out and the rate at which things are going in and out, so that things are going in and out at the right rate rather than the wrong rate. So we need to, your cell needs to have some control over the fluidity. And again, they can think of this kind of like homeostasis, maintaining a homeostasis, maintaining the right level of fluidity helps maintain the right level of permeability. Okay, so fluidity is closely related to the phospholipid structure. We also need to talk a little bit about membrane proteins. We've talked a lot about phospholipid structure, but we haven't talked a lot about the proteins in the membrane. So this is showing you an example of a membrane protein. And I forget, honestly, what name of a protein it is. There are different forms of proteins. We'll start with that. So there's two different categories of membrane proteins. So there are ones that we call integral proteins. What do you think we mean by integral? Yeah, it's inside the axial membrane. So this would be an example of an integral protein. It looks like a kidney bean to me. So it's within the membrane, within the structure itself. Some of these are what we call transmembrane proteins. Meaning what? Trans goes all the way through. Like it's things that can go, passes all the way through the membrane. So this would be a transmembrane integral protein because it's sticks out on both the extracellular side and the cytoplasmic side. It's going all the way through. There are other ones like this one that are also integral, but they don't necessarily stick out on both sides. They don't have a fancy name. So they're just partially embedded. So they're partially embedded in the membrane, but they're not sticking out on both sides. So this is a zoom in of a transmembrane protein. And you can see some of its structure here. So this secondary structure, what secondary structure does this one have? Yeah, it's got these alpha helixes that are in the middle. but you don't see them on the ends, and it's actually, this is a lot of it just stopped writing. A lot of transmembrane or integral membrane proteins are also amphipathic, meaning they have regions that are hydrophilic and regions that are hydrophobic. So like the phospholipids, they're gonna the hydrophobic regions are gonna be away from the water. So these alpha helix parts are hydrophobic, but these parts that are exposed to the cytoplasmic side and the extracellular matrix side, or the extracellular side, these are hydrophilic. So different regions of the protein have a different characteristics, again, based on things like their side chains and the way that they interact. So they're also amphipathic. So just like the phospholipids are amphipathic, the second kind of protein, besides an integral protein, you see these proteins here that are not really embedded in the membrane. They're just kind of sticking off the membrane. And they're called peripheral proteins. So they're not actually embedded in the membrane. They're just kind of bound to the surface. Sometimes they're bound to lipids. Sometimes they're bound to integral proteins. So like this one would be bound to a protein. This one's bound to the lipids, but they're just loosely bound to that surface. And it may be that they're bound to the cytoskeleton or the extracellular matrix, depending on which side of the membrane they're on. If they're on the extracellular side, they might be bound to the extracellular matrix. If they're on the cytoplasmic side, they might be bound to the cytoskeleton. A lot of times these membrane proteins are clustered in groups in the membrane. So you'll see groups of proteins and then sections without any proteins. And again, you'll see groups that have both integral and peripheral proteins on them. And you also see here, a lot of them have these tags on them. What kinds of molecules are these? Carbohydrates. So we're calling them glycoproteins. And those have a function, which I wish I had added a slide. I did not. Okay, I'm going to erase this. Sorry. Not that my handwriting is. Can I erase? Yes, all of it. There we go. There can be, we're going to talk about 6 functions that membrane proteins can have. And those glycoproteins will come in on this. Okay, so one function of those proteins, well, why don't you tell me? What do you think that these proteins can do? So one of them is actually, yeah, like communication, which we give a fancier name, signal transduction. So they are able to relay messages. Now, what kinds of messages are cells sending each other? Hey, this class is really boring. When is it over? Can't believe we have a chemistry test on Wednesday. No. This could be like hormones, which actually tests could invoke hormone responses, but. So it could be chemical messages like hormones. So it can receive the message and then bring it into the cell, so the cell knows what to do. What else do you think proteins can do? Yeah, they have transport functions. They can act as a channel. or as a gated channel, meaning they're controlling what goes in and out, not just letting it freely pass through. But something can come through the membrane either way, in or out of the cell, through a protein. Chapter 8, we'll talk a lot more about this, but these membrane proteins can also function as enzymes. Enzymes help catalyze the reactions that are really important to cells. And so sometimes these enzymes are actually embedded in the membranes, like we talked about with the mitochondria or the chloroplasts. Some of the enzymes that are necessary for cellular respiration and photosynthesis are embedded in those membranes. So those membrane proteins can function as enzymes. Another function we just talked about a couple slides ago, okay, very end of chapter 6. What else can proteins do for cells? We talked about 3 different kinds of cell junctions, because proteins can help to, connecting cells, holding them together. They provide that connection to make them into tissues to function together. They also can attach, so that's between cells. They can also attach to the extracellular matrix. So they can attach the cell to the extracellular matrix. And last but not least, they can do function in cell-to-cell recognition. And this is where those carbohydrates that are attached function like tags, like labels to say, hey, I know you belong here. We've talked about blood typing ever, you know your blood type, A, B, B, O. The blood types come about because you have specific kinds of glycoproteins on the red blood cell membranes. So I have type A blood. If I get type B blood as a transfusion, my body's going to say, hey, wait a minute, that doesn't look like a tag that belongs here. Let's do something about it. Oh, I'm dead. Okay. Maybe not the best response, but it's recognizing the cells don't belong there. But those cells, those carbohydrate tags are there for the recognition. So lots of different functions of the proteins. We talked about membrane fluidity and how fluidity affects membrane permeability. And we will finish with this slide, but we're finishing with this slide, so we're not done yet. This is possibly the membrane structure is determining permeability, and this is the most important function of your cell membranes, is getting things in and out. And so it is selectively permeable. What does that mean? It has control for the most part about what goes in and out. Okay, I think of it kind of like a colander, except that this is not a great analogy, but I feel like you're struggling with anything I was hands-on today. If I'm making pasta, what do I want? The pasta, what do I not want? Water. So this is selectively permeable, assuming my pasta is bigger than the holes that are in the colander. I get to keep the pasta and I don't keep the water. One time my mom made mint tea, where you just like boil mint leaves in water. She was so proud she made it for my dad and used to have it growing up. And then she took the pot of mint tea and she dumped it in the colander. And she kept the mint, but not the mint tea part. Because we're not used to keeping, usually you don't keep the water part. It was really funny at the time. We laughed at her a lot. So selectively permeable being the idea of controlling what goes in and out, because it's a two-way street. It's not just it's not just that things need to come in, things also need to go out. So what are the kinds of things that a cell would want to come in? What do cells want? What does the membrane need to let in? Nutrients, so things like sugars. We need sugars for cellular respiration for energy. So the cell membrane has to be able to allow sugars in. What else? What do cells make in the ribosomes? Proteins. So what do they need to let in to be able to make proteins? What? What are proteins made out of? Amino acids. So there has to be a way for amino acids to get in. You can think of this also just as raw materials in general, not just amino acids. One more thing that your cells need. Nutrients, raw materials. As you're sitting here, what are you doing? Drinking water? So you might need to let water in. Okay, I'll give you two more things. What else are you doing? Yeah, you're breathing because you need to bring oxygen into your cells. So the cell membrane has to be permeable enough to let things like sugars and raw materials and water and oxygen in. And on Wednesday, we'll talk about what it needs to let out and how it does these things. What do you not do?